Which of the following is seen in the telophase stage? Number 1, chromosome cluster to opposite spindle pole. It is right. Chromosomes moves towards the opposite pole. That is, suppose this is a cell. Chromosomes, they get separated in anaphase and moves towards the opposite pole. At the end of the anaphase stage, that is beginning of the telophase stage, nuclear member and nuclear envelope and nucleolus, now they reappear. Golgi body, Golgi complex, they are formed just reverse of prophase. We can say telophase is a reverse of prophase. Reverse of prophase is telophase. Reverse of prophase is known as telophase. So all these events are seen in telophase stage. That's why the answer is more than one. Answer is more than one. Term meiosis, information based question, answer is former and more. Term mitosis, Walter Fleming, whereas term meiosis was given by the former and more. Meiosis is significant. First one, meiosis is actually reduction division. Meiosis is actually reductional division, or it is also called reduction division. Meiosis is known as reduction division. Why it is reduction division? That is, if a cell has 2 and 4, in meiosis it divides to form 4 daughter cells, and each one with the half of the number of chromosomes from the parent cell. Each one with the half of the number of chromosomes from parental cell. Each one with the half of the number of chromosomes from parental cell. If these two cells fuse, then again that number is restored which was in the parent cell. When these two cells fuse, then the number is restored. So that's why the significance of meiosis, as far as significance of meiosis, identical cell production, wrong answer. Restore the original number of chromosome after fertilization, it is the right answer. There is a doubling of DNA contained in the cell, it is once, but that takes place in all division. Mitosis as well occurs only in vegetative cell, this is wrong. So all these are wrong except this one, that is, the due to meiosis this number is restored, because in meiosis, after meiosis those cells are formed. If they are gamete, they fuse together to form a cell, that is zygote, and the number of zygote, number of chromosome as well as genome is restored in the zygote, as it was in the parent cell. Which of the following is not the characteristic feature of meiosis? It involves 2 stages of DNA. Yes, no, DNA duplicates only once. That is, DNA duplicates once. No DNA replication 2 stages. That is, suppose a cell has 2n is equal to 4 and it has 2C DNA. In interphase stage, number remains same, or number of chromosomes remains same. That is, 4 chromosomes are there, but the amount of DNA is 4C. This is the replication of DNA, followed by first meiosis. In first meiosis, 2 cells get 2C DNA. Then in second meiosis, these 2 cells receive 1C DNA. If we compare the content of DNA from parent cell, it is reduced to half. But as far as replication concerned, DNA replicate or duplicate once and that is in the interphase stage before meiosis 1. That is in interphase stage in meiosis 1. It involves recombination and crossing over right. Sister chromatids separates during anaphase 2 right. Nuclear membrane disappear during prophase right. So, which one is not correct? That is 2 duplication of DNA. DNA replicates 1 in any kind of type of cell division. DNA replicates 1. For meiosis, you can say DNA replication once, DNA replicates once, whereas cell divides twice. Cell divides twice and 4 daughter cells are formed. 4 daughter cells are formed. And in these daughter cells, number of chromosomes reduced to half. That is, suppose cell has 4 chromosomes, then you will see that 2 chromosomes in daughter cell, 2 chromosomes in daughter cell. These are the some features of meiosis. 27. Prophase of reduction division. Actually, in meiosis, cell divides twice. 2n is equal to the 4. In division 1, number of chromosome reduced to half. This is first meiosis, and meiosis 1 is reductional. Whereas in second meiosis, number remain as same as it was at the end of first meiosis. That is, meiosis 2 is mitosis like. Meiosis 2 is mitosis like. So, meiosis 2, that is equational division. Meiosis 2 is known as equational division. In meiosis 1, it is full of events. First meiosis comprises of 4 phases: prophase 1, metaphase 1, anaphase 1 and telophase 1. These are the 4 phases, telophase 1. And prophase 1 is long and eventful. It can't be seen in other type of cell division. Long and eventful, comprises of 5 phases. Prophase 1 comprises of 5 phases, and these 5 phases orderly manner, that is, start with the leptotene, second is zygotene, third is packetine, fourth is diplotene, last is dikinesis. This is the correct sequence. First leptotene, second zygotene, third packetine, fourth diplotene, and the last is dikinesis. Last one is dikinesis. Crossing over. Actually, each stage of prophase 1 has some speciality. Prophase 1, I have already mentioned, comprises of 5 phases. First is lactotein. In lactotein, chromosome appears. That is, suppose this is a cell and this is nucleus. There is gradual appearance of chromosomes as a result of condensation and dehydration. One more speciality of lactotein, that is, chromosomes are attached to the nuclear membrane from both the ends, either in this manner or in this manner. Attachment of chromosomes from their both ends to the nuclear membrane. This is bouquet formation. This is seen in lactotein. In zygotein stage, pairing of homologous chromosome takes place. That is, homologous chromosome forms pair. That is zygotein. And pairing of homologous chromosome is known as synapsis. Third is packeting. Each chromosome splits longitudinally. So there are two chromosomes, that is, pair is there. Pair is called bivalent. Pair is known as bivalent. And each member of bivalent splits. So each chromosome have 2 chromatids, total number of chromatids 4. This is tetrad stage. Tetrad is formed. Tetrad formation packeting. Followed by crossing over. What is crossing over? These 2 chromatids of a chromosome are called sister chromatids, and these 2 are also sister. But there is a segmental exchange between non-sister chromatids. Segment of a chromatid of non-sister chromatid. Yeah, that is non-sister chromatids. There is a exchange of segment in between them. That is known as crossing over. So 2 events, that is tetrad formation followed by crossing over, that is feature of packeting. And in packeting, 2 non-sister chromatids of bivalent are involved. 2 non-sister chromatids. Let's check again. That is, what is non-sister chromatid? This is a chromosome with 2 chromatids. And these 2 chromatids are called sister chromatids. Now this is also a homologous chromosome. This is homologous chromosome of the previous one. These 2 are also sister chromatids. This is pair of homologous chromosome. These 2 are the sister chromatids, and similarly these 2 are also sister chromatids. But at the time of crossing over, one particular segment, suppose this one, is exchanged with this one. That is, segmental exchange takes place between 2 non-sister chromatids in a pair of homologous chromosomes is called crossing over. Segmental exchange between 2 non-sister chromatids is known as crossing over. This term was coined by T.H. Morgan. This term was given by T.H. Morgan. Gave the term crossing over. T.H. Morgan gave the term crossing over. And that's why there is a shifting of allele from one chromatid to another one. This is recombination. This is also a kind of a recombination. Pachytene stage of meiosis, chromosomes appear. Chromosomes appeared, each chromosome with 2 strands. We can say each chromosome with 2 strands. And total in bivalent 2 chromosomes are there. 2 chromosomes are there. So 4 stranded stage is obtained. 4 stranded stage is obtained. As far as chromosome concerned, if single chromosome, then it is double stranded. But if we consider the pair of homologous chromosome, then 4 stranded. But for each chromosome, if we write here each, then each chromosome is 2 stranded. Each chromosome is 2 stranded. But as far as bivalent concerned, if bivalent is mentioned, then 4 stranded stage is there. So here only chromosome word is written. Here chromosome word is written not as bivalent. So we can say for each chromosome that is 2 stranded. This is 1, this is 2. Crossing over enzyme mediated process. Group of enzymes which are required for the crossing over recombinase. It's already question for the NEET. This is for the NEET question. It is already asked in the NEET examination. Group of enzymes are required for the crossing over. That group is termed as recombinase. Gyrase is required by the prokaryotic cell at the time of replication of DNA. Polymerases are also involved in the replication of DNA as well as RNA. Hydrolases are enzymes contained in the lysosome. They are responsible for the digestion only. Repulsion of homologous chromosome takes place. Repulsion of homologous chromosome. See this one. In pachytene stage, crossing over takes place. But in diplotin stage, these homologous chromosomes repel each other. In diplotin stage, there is a repulsion of homologous chromosomes and they are attached only one particular place. They are attached at the one particular place where the crossing over occurred. That X-like structure is known as chiasma. That X-like structure is known as chiasma. So, chiasma is seen in diplotin because of repulsion in homologous chromosomes. So, here the best answer is diplotin stage. Visibility of chiasma 1 and second feature dissolution of synaptonemal complex. Diplotin is known for visibility of chiasma. Chiasma is visible and second dissolution of synaptonemal complex. Dissolution of synaptonemal complex. The answer is clear. In the previous question, I have already mentioned Answer is diplotin stage. Next one, terminalization, that is termination of chiasma. Position of chiasma is at here now. Then gradual separation takes place. Now see the position of chiasma slightly shifted. Then see the next stage. It moves towards tip. Then almost at the end, and finally get these two non-sister chromatids get separated. This is known as terminalization. This is known as terminalization and is seen in dikinesis. This is the last phase of prophase 1. The process of terminalization, that is termination of chiasma. What is terminalization? That is termination of chiasma. Termination of chiasma is known as terminalization. In oocytes of some vertebrate, it's an informative question and NCERT based question in cell cycle. You can clearly see, and the answer is diplotin stage. The answer is diplotin for 34th question. 35. This stage is represented in the diagram. See the chromosome. Each chromosome with one chromatid. Each chromosome with one chromatid actually. That is, if it is with one chromatid, it is with the one chromatid. That means the diagram is of anaphase stage. Diagram is, anaphase 2. Because two chromatids are seen in anaphase 1 in meiosis. This diagram is actually of anaphase stage 2. Anaphase stage 2, chromosome when they move, there is a division of centromere, and anaphase 2, there is a division of centromere, followed by their separation. At the time of separation, they have only one chromatid. And that is indicated in the diagram. So this is anaphase 2. The number of chromatid in each chromosome at anaphase: 2 in mitosis, 1 in meiosis. Depends at which stage is asked. 1 in mitosis, 2 in meiosis. 2 in mitosis, 4 in meiosis. The number of chromatids in a chromosome. In anaphase stage, normal anaphase stage, actually there is a division of centromere. So each chromosome with one chromatid. Each chromosome is with one chromatid. The one in mitosis. And for meiosis, actually at anaphase 1, if we discuss about the anaphase 1, then in anaphase 1, 2 chromatids are there with chromosome, that is at anaphase 1, whereas anaphase 2, that is each one with one chromatid, This question is not quite clear. The number of chromatids in a chromosome at anaphase. So better answer we can take the any one particular cell division, and that will be better. That is, if we take only one chromatid in mitosis, this is sufficient, rest discarded. We should replace this question. Okay. Second division of meiosis, that is equational division as same as mitosis. It is same as mitosis. Mitosis like division. That's why it is called equational division. It is mitosis like. Mitosis like because at the end of meiosis 1, if cell has 2 chromosomes, then at the end of meiosis 2, these number is same. So it is mitosis like. It is mitosis like, and mitosis is called equational division. Number is same. That's why it is equational. My answer is equational. Decision for the cell division, that is in G1 stage. That is clear answer is G1 when cell crosses its first checkpoint, and that checkpoint is in G1. When cell crosses first checkpoint, that is R point, there is a first checkpoint and major checkpoint. If cell crosses, then cell will definitely divide. So the decision of cell division takes place in or occurs in G1 phase. A plant species 10 picogram DNA and 8 chromosomes in its pollen grain. Pollen grains are haploid. Pollen grains are haploid. Amount of DNA is 10 picogram and number of chromosomes 8. This is for the pollen grain. And pollen grains are formed as a result of what? That is meiosis. In G1 stage, G1, S and G2 stage, G1, S and G2 stage. At G1 stage, 8 chromosome plus 8 because cell is diploid. That is 16 chromosomes are there in pollen mother cell. If the number of chromosomes is 16, then definitely the amount of DNA should be 20 picogram. Amount of DNA should be 20 picogram. For 8 chromosome, if for 8 chromosome 10 picogram, the chromosome number is double in the parent cell, and at the G1 stage it is not duplicated. It is not duplicated. So just double the number of chromosome and double the amount of DNA. So my answer for this question is 20 picogram DNA and 16 chromosome at G1. 20 picogram DNA and 16 chromosomes at the G1. When the cell crosses G1, that is in S phase, the amount of DNA is doubled, but the number of chromosomes remains same. This is till the G2 stage. In meiosis 1, then 8 plus 8, 2 cells are formed, then followed by the second is same as the first one, but the number of chromatids reduced. Select the event which occurs twice during meiosis. Nuclear division, yes, two times. Division of cytoplasm 2 times. DNA replication only 1. Bivalent formation only 1. Equational division only 1. That's why my answer is only A and B. These 2 events takes place twice. That is division of nucleus and division of cytoplasm takes twice. DNA replicates once, bivalent formation once, that is in prophase 1 of meiosis. Then equational division, that is second division is the equational, not first division. 41st question. Select the correct sequence of event. 41st question. Select the correct sequence of event takes place during meiosis 2. First, condensation of chromatin, right. Splitting of centromere, right. But prior to, that is, first A takes place, followed by C, then B. A, C, B is correct answer. A, C, B. The condensation takes place in the prophase 2, then chromosomal alignment at equator, that is at metaphase 2, and according to sequence prophase followed by metaphase then anaphase. So this is the event of prophase 2. This is the event of prophase 2. This takes place in anaphase 2. This takes place in anaphase 2, where this one is seen in metaphase 2. Metaphase 2. So arrange them, first prophase, then metaphase, and third is anaphase. So answer is ACB. How many times the meiosis takes place for the formation of 80 wheat grain? For the grain formation, ovule is required. Ovule is needed for the formation of one grain. So number of ovules required 80, and in each ovule one meiosis takes place, one meiosis per ovule. So till the ovule 80 meiosis are there in 80 ovules. Then each ovule is fertilized by one pollen grain. Each ovule is fertilized by the one pollen grain, then only it is converted into grain. For the pollination or fertilization of 80 ovules, 80 pollen grains are required. And these 80 pollen grains are formed from the 20 pollen mother cell by meiosis. These are the PMC, that is pollen mother cell. They perform a meiosis to form 80 pollen grain, and these 80 pollen grains are needed for the fertilization of 80 ovules. So in that case, meiosis of 80 ovule and meiosis of 20 pollen mother cells, that is total 100. Total 100 meiosis takes place. Or there is a shortcut, that is number of grain or seed n plus n by 4. You can apply this. This is the number of ovule, this is number of PMC. Pollen mother cell, number of pollen grain divided by 4, you will get the number of PMC. Number of pollen mother cells can be obtained if we divide the number of pollen grain by 4. Each PMC produces 4. So here 80 grains are required, the value of n is 80, then again the value of n is 80, 80 by 4, that is 20. Answer will be 100. Answer will be 100. They are the two ways by which this answer can be known. The logical, this is shortcut, this is logical, this is shortcut, but applicable for seed, applicable for grain, but not pollen grain, clearly not for pollen grain. To obtain 100 pollen grains, then divided by 4, that is 25 meiosis is sufficient. Not for pollen grain. This is for the wheat grain, rice grain, maize grain, etc. Not for pollen grain. Between mitosis, a cell is called to be the. This question is again, I don't think is complete. Between the mitosis, that is resting phase. That is resting phase. Better answer is resting phase. Why it is resting phase? Let's know. Actually, there are two phases of mitosis. First is interphase. That is preparatory phase. That is, no apparent division of cell is seen in this phase, followed by M phase. So M phase, resting phase, is actually interphase. Cell prepares itself for the division, not sleeping, not active. Metabolically active, we can say, metabolically active. But here the question is very simple, that is, cell is called, then it is in resting stage. endomitosis, that is within, endo for within, that is endomitosis, that is division of chromosomes, duplication of chromosome, but there is no division in the nuclear membrane. That is, duplication and division of chromosome takes place but inside the nucleus only. That is known as endomitosis. And the final one, the term mitosis. Term mitosis I have already stated when I have started the discussion, that is W. Fleming. W. Fleming gave the term mitosis. Mitosis is also called somatic cell division. Mitosis is also known as somatic cell division. Somatic cell division or equational cell division. Equational cell division. Other two term for mitosis. Other two term for the mitosis. That's all for the cell cycle. Thank you.